will just introduce you briefly, Yogesh.
Yeah, I think we also have a time, so I think just we maybe I can be like introduce myself also that you don't need to.
So I will just in one phrase
Okay.
telling to audience here that I met Yogesh like years ago when I was postdoc in Tübingen in Germany, and I found in him a wonderful and very amazed researcher with a lot of ideas. And now he is established as a group leader. He got a lot of found successful research and allowing him to establish himself as a group leader. So group leader is an independent researcher with his own lab, with his own academic research idea, and he is doing mainly a lot of stuff on neurodegenerative disease. And today he will tell us more about the link that exists between gut microbiota and brain disease. This is what I have to say. And you also are welcome to introduce more yourself academically and start your presentation. Again, thank you very much, Yogesh.
Thank you, Rocco. I think I don't need to introduce myself more. I think you already explained so many things. So I think it's very kind of you. And I'm really privileged to have like your invitation for this talk. So I think that's a talk that I made, especially for the students as well. So because I wasn't sure like how much students know about microbiome. So I will go as my teaching because I also do a lot of teaching on the microbiome. So I thought this could be like kind of a good kind of a mixed. And then I will go to about my research that what I have done and kind of what we are kind of doing. So, since 2020, I have my kind of a more kind of independent group, and then we are working on the microbiome. And so this is what also in Parkinson's disease and especially in Huntington's. And so we have 3 different models that we are working right now. And then I will go through slowly by one by each, and then I will try to like communicate and then teach you something about about how the gut microbiome can affect the different neurodegenerative diseases, especially Parkinson's disease. So the talk is actually, it's like I will talk a bit about how the basic definition of the microbiome. Atif, like he's already already talked about earlier. So, but I thought I can just talk to you a bit more how the microbial genomics and they can be detected. So this is beautifully described earlier in the talk. I'll just summarize again, and how the microbiome actually improves or keeps our health in check. This is kind of very important aspect, not only like a neurodegeneration, but in all different kind of diseases. Actually, so far, there's a lot of research has been done in the different fields, such as diabetes, metabolic disorders, infection, and so on. So that many different all disorders that are actually affected by microbiome. And especially like talk about the microbiome and different neurodegenerative diseases. And then I will summarize the talk, and then I will happy to take any questions afterwards. So because it was talk for the like I teach the students, so I will see like, okay, what you see is actually on a slide, because normally you cannot see the microbiome, but when you see under the microscope, then you can see all different kind of like a bacteria, just lactobacilli, E. coli, bifidobacterium. And so this is kind of a very kind of a general kind of a bacterium. You see different shapes, different size, and that's how you can, but normally you can see because these are the culturable bacteria. So then you can culture them, you can see under the microscope. This is when traditionally has been kind of described. But since the genomic tools started to improve, and then we can detect all the genome, like DNA of these bacteria, or then we started to really understand all the different microbiome, especially like a gut or skin or so on. So this is just to give you a bit more kind of, again, the definition. What can call the microbiome? The microbiome, the assemblage of microorganisms, which contains the bacteria, archaea, and eukaryotes and viruses. So this is like a mixture of all different things. So normally people kind of understand like microbiome, that people always think about bacteria. But in general, like people are focus a lot on the bacteria rather than any other like archaea or eukaryotes and viruses. So, and there's a special defined environment which can say like, oh, this bacteria or microbiome environment is makes the like a specific group of like the specific group, which you can say like this is from the gut or definition or gut or skin and so on. So the term microbiome is perceived by the name of the environment in which it is located. So such as gut microbiome refers to the microbiome in the intestinal tract, or skin microbiome, or also on different body like organs that we can have like oral microbiome in mouth cavity and so on. So the microbiome is a community used to refer the microorganism you harbor in and on your body. So this is just a simple to the audience which actually have no kind of previous kind of a prior knowledge of the kind of a microbiome. So microbiome acts very interesting because they are like a lot in numbers. So this is so normally initially it was thought that there was more microbial than human cells in the body. So that normally people used to think there's a 10 times more the microbial cell means the amount. But now the recent estimate has been changed, which now normally says that human microbial and human cells are present in almost very comparable numbers. So it's not like a tenfold more or not, but this is kind of where this an individual weighing around for 70 kilos, which is the human microbiome also very more than around 100 trillion microorganism, and they also weighs around 200 grams. So there's like around 150 to 200 times more genes in the individual, which comes derived from the microbiome compared to the like a human sort, because now human but we have more kind of a genetic makeup from the micro. So actually this itself tells us that the microbiome is kind of very important, why we need so much genome, and so then can affect our health. So normally human genome consists around 20, 20, 20, 20 to 20,000 genes, which are like, whereas microbiome has over more than 3 million genes. So which can produce thousands of like a different metabolites. In other words, we say the genes that we have like a human, like the microbiome contains more than 99% compared to the human. So it's like a lot, which is like, this is how we kind of evolve during the evolution. Like if you think about the like evolution, how the microbiome evolved and then the human came later. So this, we always live in a kind of that kind of symbiosis. But important aspect is the core microbiome, which is like found in all healthy humans. And with this core microbiome start to reshuffle or change, then we started to have problem like mostly the disorders and so on. The what factors can influence the microbiome? This is normally now I will bit more focus on the gut microbiome because I work on the mostly in the gut. So the factors which like kind of like human can take actions like a feeding methods such as breast milk, artificial milk, and introduction of solid food. So then the baby is born in early life, then they have a different kind of like feeding. So it depends on feeding the microbiome also, and the health of the baby can also be affected. And then medication. So now we take like from the child, like since we born, we depends on like how sick we get. We take antibiotics. We also take some like other medical suppressant and also drugs in the when the later life. So then they can also affect actually of the microbiome, and also our dietary habits, with the way food is cooked. If we're eating more raw food, then we have a different diversity and or different kind of microbiome. And the lifestyle and environment we live, such as rural versus urban location, on exercise, this is also very important too. This can affect also the microbiome. And then like the weight gain, if we have also weight, then we can have a kind of a different kind of microbiome. The factors which human cannot control, like we cannot control or cannot take a direct action, that our genetics, because everybody is in different and different genetic makeup. Each human has a different, I mean, we have all same genes, but like depends on like your ancestry. That can be also which the microbiome means we cannot have non-control. The anatomical part of the intestinal tracts, like the large intestine can have a higher microbial diversity compared to the small intestine. So this is like the way it is, like depends on the environment, because large intestine has a more, means small intestine has a more oxygen compared to the large intestine, which is like, so that's why allowed to grow more anaerobic microbial communities. And gestational age. So because some children are born preterm birth and also full term, this can also affect the like health of the baby, and also the health like the microbial diversity later on the lifestyle. And how the baby is born, whether it's a vaginal delivery or C-section, this can also like affect the microbial community into the like of the person. And aging, of course, the aging happens, depends on each person age differently, and then can also affect your microbiome. So but we don't have a control on this direction. So we can actually say as like microbiome can act as like individual ID card, as you have your passport, that you have your information and so on. And then this can bring the unique features and function to the human. And this is what we can say, like microbiome or your unique gut microbiome can be considered as a more kind of an ID card for you. So moving on to the diet and microbiome, how the of course, as I told, we know that the gut microbiome has a genetic component, but the diet and lifestyle, environment, and then the outside, like this antibiotics, the medication can influence the gut microbial composition. And what you eat, then you can also make your gut, your microbiome. So you can reshuffle your microbiome, but always, I said earlier, the core microbiome always very fixed and stable. So we can have like diet change, but it doesn't change all within a second or day, because it takes us some time to. So it's not roughly, we can talk about more kind of 24 hours or so. Then, and the more the diverse the diet is, the more the diverse the microbiome is. So if we're eating more like diverse kind of food, then more likely our health is more. And this is that traditional knowledge that we have in the past, like we people who eat like all different diverse kind of thing, they are much more kind of healthy. And also how they you eat, like the way you eat can also affect your microbiome. So if you have a dietary fibers, mostly like if you're eating vegetables and so on, then your microbiome different. And if you're also having more probiotics like fermented yogurt, milk, and coffee, then can also increase your diversity. And the amount, like you having more plant-based proteins, and then that can also increase the diversity of your microbiome. And some people also eating fish and like avocado and so on, they can rich in diet, like a different this the omega-3 and omega-6 different fatty acid, then they can also affect. So cut the story short, like it's a different dietary habit and a different lifestyle environment can really affect your microbiome, and can ultimately can also influence your the health, and which can depends on what kind of this. So of course the genetic makeup is the more important, but it can trigger certain diseases also, like if the microbiome change, and this is why this slow kind of process. So coming back to the, I think, the normal health associated microbiome, there's no consensus on what constitutes a health associated microbiome. Some hallmarks has been proposed in the literature. So the increased species, richness or diversity, the number of different species, and how different they are in a specific habitat, such as digestive tract, is considered as a hallmark of gut health. So we can say more the diverse is more kind of healthy, but we also have some resistance, resilience, and stability over the time. So microbiome also resistant. And then this ability to put the change or perturbation with antibiotic or imbalanced diet can recover or can be a stable state. But we always depends on your genetics, can also not be recovered. So that can also lead to the different kind of disease as well. And high gut microbial gene richness, which is the number of microbial genes in the gut, has been suggested as an indicator of good metabolic and overall health. And a lot digestive elements such as obesity, diabetes, allergy, cancer, and now since 2014, the neurodegenerative diseases, because before that people always thought mostly the neurodegenerative diseases are based on genetics. This we cannot have a component of the our health, our gut. And but recently, since 2014, we started to have a lot of studies published in this published, and also kind of used to understand how this the microbiome actually has a key component, not entirely affecting, but it's one of the key component which is modulating the disease function as well. So this is, and any change in the imbalance in the gut microbiome considered as a dysbiotic or dysbiosis. So this is kind of this is another this is the potential way of dealing with the many different chronic diseases, because such as inflammatory bowel disease, which we have a very severe change in the gut microbiome. And because we have also a lot rise in the Western society, especially in the Europe and also in America, because the diet, the way we are living is changing our microbial community. So just give you one example, so the developing countries such as India or China, they are developing really fast, and their diversity is kind of severely declining. And then when we talk about Europe and America, then they have always a less reduced overall diversity. But when we think about the countries like Latin America, like a Peru and so, and then the study was done, and then they identified the microbial diversity actually in the human population or also the native like a population where it's like they are not intervened by this like different fancy like a diet, and so when they're living in a natural lifestyle, they have a huge microbial diversity compared to the diversity we have in the Western or maybe American kind of population. So this tells us like having a like diverse lifestyle, and then and the environment, and not only like lifestyle but environment can also really play an important role. So then and the countries who are very much developing very faster, they have a very kind of rapid decline on the population of the microbial diversity. So the coming back to why microbiome is important. So like, so of course, there's like many functions which can like we have like consider like a defense. So it defend against different harmful microorganism, and also like teaches the immune system to tell whether this is like a friends or foe, because the different bacteria community is growing in the stomach, and every day like we getting exposed. And our immune system is like, because blood cells normally means I have a bit more immunology background, so I can tell you a bit more about immune system, because since the baby is born or child is born, the like this is naive, this immune system doesn't know anything. Over the time we have like a training of the immune system, because this is internal, outside, and this is how it start to differentiate. Okay, this is something we have to like get a tolerance about the like our immune system, and maybe it's like pathogenic, then we should eradicate from our body. So this is how the microbial community also train our immune system regularly to tell, okay, the body, okay, this is really needed, this is not. And also like microbials, if they have like toxic compound, they can also degrade these, because they have very potential metabolic activity. So they can make so the drugs you take in something, they can also be discarded. And the nutrition, so like of course this like helps them like a dietary fibers, because the human cannot digest. So that's why we always need this microbiome, and so that's why we start to feel like later on when our microbial diversity start to change, then we can also have some gastric issues because of like the diet we take, and that cannot be like digested when some of the bacteria which are feeding on the dietary fiber. So some people, this I can give you one example, like when some people say like lactose intolerance. So that means there's one study is done because they cannot have those subtypes of bacteria which cannot really take the lactose, and thus we cannot digest, and that I think that's why we started to have problem. And also like absorption, so dietary like not only like it helps to absorption or dietary minerals as well, like magnesium, calcium, and iron. So because they can also synthesize like such as like vitamin K, folate, and amino acid, which is the building blocks of the protein. So in summary, I can say like then also like they can also change the like how the like our shape, like mood and behavior. So then this bacteria, like the microbiome, not bacteria, they are involved in like the one study has been done like a few years back in the in the Belgium, and then they have taken the like almost more than 1,000 students who come to study in university in bachelors from year 1 and year 3, and then they identify almost like a 20% of the students feel like this the depression, and the depression is like linked with the like some inflammatory bacteria versus LSTP. So this is one of the like example I can give you, and I talked about this bacteria, then I will talk in our study that what we found is something very kind of similar. So this could be like a really involved with the mood and also behavior, how this like kind of affect our behavior, because they can also eat like certain like the means active compounds like this dopamine. So if their dopamine can be affected, then of course this compound can change our mood and behavior. So I think I will not dwell too much. This is just want to talk about how the different methods to attack the microbial functions. So who is there, which is you can really understand the microbial potential available under cellular materials. So we can ancient methods that microscopy is still the gold standard, and then culture. So all the bacteria and the microorganism cannot be grown, and so that's why we have to have this like a DNA based tools. And the second thing is what can they do? So that of course they have a lot metabolic potential. So then based on the genetic material, we can identify what kind of a functions they have and what kind of a network they can have. And not only like one function, because community this lives in a network. If you take out one community bacteria, then it can also like imbalance the community. So this is why having a community is very kind of important. And this understanding not as individual bacteria, but also a community is very important, like the theme of the research right now we have, like trying to understand that. And what are they doing? So this function is such as metabolic pathways, and that's, so this can be also understand the protein level and metabolite level, which we can do the different, as previous talked about the meta like transcriptomics. Also, we can also do, like, now we start to have more tools. It's still not very great, but still it's the good thing. And the metabolomics has already been like implemented in many studies. So mostly like study has been done on a DNA basis, and also the metabolite label. So because these are the final out product of the like your for your gene or DNA, and then we can really understand how this like this they interact, and then we can predict the functions, how they're going to be behaving in a particular setting or disease. Means, of course, the earlier presentation talked about the cave, because this is like really fascinating, and I think the same we can think of like what is happening inside our body as well. So just to give you a very brief kind of a, like how that like in the lives, like different life stages, our like microbiome kind of changes. So first 1000 days of the life is kind of very important, because here like you have most of the kind of no like a community of the bacteria or microbiome. And then you start to develop. That's why if you think of like the early children when they're young, then they start to have a colic pain because their colon is kind of clean, and then we eating, all children start to eat, and then they developing this like community of the microbial. And that's why they have all this like this like a flatulence and gas and so on. And this is because the bacteria also trying to establish themselves in the gut, and slowly making the environment like so they can live in there. And in the early like high richness, mostly the most of the our like uh, means the microbiome is consisted of Firmicutes and the bacteria and the phyla. So these are the most two major dominating phyla we find in the human gut. And our microbiome in adulthood is very kind of a consistent from like until 40 or so on. When people start to get more elderly, then we have a lower diversity and enrichment of the more potential the harmful bacterial groups. So this is kind of this we have a very good stable, and when we progress, then our diversity is kind of a decrease. And when we initially we have no diversity, because there's also some contradictory finding that in initial like in the stage, we some people say in the uterus is like has a microbiome, but some says no. So it's always still contradictory kind of findings. So not to go on that side and then move on to the further. So this is just to give you again the repetition of the same kind of slide, but here just wanted to like talk about that in early life we have aerobic bacteria, then we move more to anaerobic bacteria. So mostly we have enterococcus, staphylococcus, and then later we dominate by mostly the Firmicutes and Bacteroides in the adult life. And of course, the infant feeding and diet, environment is all can affect, and then can also like prone to make in the when later with that like when we are like not able to control our genetic genes, then they can also lead to the different kind of disorders. So this is actually, I really like the kind of slide, like, I mean, I've taken from the like online tools, because I mean, I was a bit lazy to make the slide, but I mean, this is just wanted to like, as we talked about cave, because we all like made like live in a different environment with the different structure, and also the cave also like the different. So when we talk about the leaf, so we have like a kind of like a different like phyla, like mostly we have proteobacteria, and then you can also find also in the mud like more proteobacteria, which is like considered mostly the pathogenic bacteria comes in this particular group, such as E. coli, Shigella, and so on. They're part of the, and then we have Firmicutes. So initially, like in the plant, we normally dominated by mostly like a proteobacteria. And then we in the like a pigs, then we have more kind of different places, can have a different kind of Firmicutes ratio. And when we talk about ruminants, and then we have mostly like kind of a, okay. So this is what we kind of, sorry. So this is what, so I will not talk I will not talk too much about this, the previous. So this is like a different method. So I already talked about, and then keep going to move further. So I think sequencing is not so important. And this, I just wanted to talk here, this like for a few seconds here. Normal, like we have our like a blood-brain barrier in the brain, and we have a gut. So we also have the normal intestinal barrier. So when we have like a core microbiome, and if this is intact, we can have a normal like neurotoxic substances such as bacterial amyloids, LPS, and the microbial like molecules. Then if this is all intact, then we have a no cognitive impairment. But if we have this change and increase in this neurotoxic substance, that which can make the gut leaky, and also can make the leaky blood-brain barrier, which can lead to the like a cognitive impairment. So this is just kind of based on the like now the recent like the understanding we have. And this is just now in especially when in case of the inflammatory bowel disease, like we have a donor, like a healthy donor, we can also transplant the feces, and then we can kind of like improve the diversity of the microbiome. But we don't know, is short term is kind of effective, but long term and like role of microbiome is still like not known. And now people also thinking about synthetic microbiome, like how the different defined structure can be like used, but it's still like there's like many like things to understand how they competing for different resources, how they can affect the long-term kind of behavior. So now it's coming back to the like means the different neurodegenerative diseases, like what has been done, mostly big, and they're taking the feces from the like different disorders, they inject it in the mice model, and then they have a cognitive impairment. That's how we came to know Alzheimer and Parkinson and how the disease can be affected. And similar, like when you have the cognitive impairment, and if you transplant to the wild type animals, they can also get cognitive impairment. So we have like a all like a mouse and like a human-based study, which like tested in animal models, which really points out that okay, the microbiome is actually one of the component which can affect the mood, behavior, and so on. This is like one of the like example. So then I will slowly move on to like what actually have been done. So since 2000, which seminal paper came in Cell, and which actually started to talk about like which normally we have a typical microbiome, which makes this the short chain fatty acid metabolites, and this can activate the microglia, which can lead to the development of Parkinson disease. But if you keep these animals in the germ-free condition, then you have no reactive microglia, and which can lead to the limited pathology. And so the last experiment was done actually how the PD-derived microbiome, if you have the patient, if you put into these animals that I talked earlier, then we can have enhanced motor functions. So that same time, actually, we have also started, I have started this microbiome project in 2016. So they already kind of published. So then we start to really understand, because what we didn't know, like how the microbial evolutions happen. So what we actually have done in our study, so we have taken the, I mean, the heterozygous mother and heterozygous father as well, because we want to have the same mother, because mother's microbiome can affect the overall microbiome. So we taken all the heterozygous, so we taken the wild type, and like this is a like a wild type and transgenic, and then we like again like have this control itermate for like different time points, and we wanted to understand in the same animals over the time how the microbial community can be changed. So as you can see in the like normally in the beginning we have this yellow is a firmicutes and red is bacteroides. So this ratio is always dominating in the gut. But over the time, this ratio is kind of start to change, and we get more firmicutes than bacteroides. And but in the case of transgenic, this ratio increase more than the wild types, then we get more dominant of firmicutes. So when we looked on this Firmicutes bacteroides, I mean, this is not really like now considered the gold standard, but we can see from the early lifestyle from the like month of like a 2 months, which like when the animals get like a fully adult or mature. And then when we think about later on this, like in the older animal, this ratio is kind of almost changed. So this means tells the microbial community in the older animals kind of change. So this is like a fits with the idea like, okay, over the aging, the microbial community fits. Well, what it tells us later on, like how the different bacteria can be really understand, like changing over the time. So we took the example of two different bacteria, one lactobacillus and one we call LSTPs, which I talked to you earlier about. The LSTPs could be really important in the mood and like the mood behavior and changes. So when we look in the yellow one in the wild type, there's no like kind of changes like, okay, like it's obviously dynamics happen in the wild type and also similar like LSTPs. But when we look in the like transgenic animals, the trend is almost like up to 2.5 months, like in the when the animals get adult. The LSTPs, which is considered like we believe like it's a more pathogenic bacteria and we can change the behavior. And the lactobacilli is kind of a down regulator. So this is like gives a very clear indication like there's a microbial community changes, and this stays here like around adulthood is like leading to some changes. We really don't know what changes can happen, and this is leading to this like, so we consider because maybe these animals are transgenic for the human gene we have in this about animal model. So mostly likely is a prone to like changes the microbial community, and that can lead to the increase in this the microbial changes. So later we looked also the how the aging also affecting the the metabolites with aging. So I mean, with aging, we can say like there's a change in the certain like lactate increase and so on in the feces. And when we look in the serum, we also see the similar, like we have increase in succinate, glutamate, and lactate, which is very similar like we found in the feces. So these are like two metabolites are really common into the like that with aging, the animals like this is kind of increasing over the time. And then when we talked about later on, like when we talked like in the serum, like how this like between the transgenic animals increasing. So we identify the succinate, which is one of the metabolite, could be increased over the time. And tryptophan, which is like also the like and tyrosine, which is the like a precursor of the like dopamine and this, so then they kind of decrease. So then we with I mean tyrosine was not significant, but tryptophan is more kind of significant. So this is the precursor of like neurotransmitters, and then these kind of decrease. So this tells us like when animals get aging, somehow these transmitter like maybe the eaten up by the bacteria or something is ongoing, which could be leading to the like change in the behavior. Well, what happens when we treat these animals? So we did the like 2 weeks treatment with antibiotic, and which can decrease this alpha-synuclein protein, which is the hallmark of the like expression for the like the Parkinson disease. So I will not talk too much about all these different figures. I think this is not important. The figure important is here, this one. So when we look this alpha-synuclein expression, which is the hallmark protein for the Parkinson disease, when we give antibiotics for 2 weeks, in like the transgenic animals, they have a reduced expression of the alpha-synuclein. So this tells us if we can change the microbial community for a short term, maybe we can like help for a short-term basis for Parkinson disease, but this is just like animal model study. So I will take always as a precaution. And then but this tells us like, of course, the microbial community is kind of very important. So the coming back to like summary, just like I try to give a bit kind of a brief kind of background about the microbiome and health maintenance. And then of course, I didn't talk too much about the how to quantify the microbiome. And especially like I talked about with more kind of Parkinson disease, which we have developed and recently published this year, how this microbial community changes over the time, and how if we change the microbial community, we can actually improve the health of this like animals, and then they can feel a bit better. So here, like I will summarize this the thing, and then now I'm really thank you for listening me. I know a bit over my time, and I'm happy to take your answers, sorry, questions, and I'm happy to answer your questions.
Thank you very much, Yogesh.
Sorry, with just teaching always.
We have here a question from the audience.
Mm-hmm.
Actually, I have two questions, but I will keep them for the end. So, go ahead.
Thank you, Yosh. Nice talk. Just have a couple of questions. The first one, what do you think about there is a new review published in Nature about, because you introduced certain numbers of cells and I would say the weight of cells and the weight of humans and so on. What do you think about this? There is 6 months ago publication, they challenged the textbook about microbiomes. Anyway, this is the first question. And the second question, in the gut microbiome, what do you recommend to go to sequence amplicons? If we see reduction of alpha diversity associated with certain disease, we can go ahead and we do shotgun sequencing, or we go directly to the shotgun sequence. This is my question. Thank you very much.
So the first question, actually, I have not read the like the paper, so then I can't really comment right now. Okay. So I have not. So then this is what I will say because okay. The second question that I can answer, because second question is most likely, I think I would really prefer to have the shotgun sequencing, because then you can have a more kind of output, because you can understand the bacterial functions. And also we can go to more like species level, because normally you have like a phyla level. You cannot go much more deeper. So I think shotgun sequencing is the way forward to really understand the.
And how many samples do you recommend, and the depth of sequencing that you recommend?
Oh, depth of sequencing. Okay. So normally we do 50 million reads, because you have 50 million. Yeah.
Shallow sequencing. So
No, like deep sequencing. But shallow is like 20 million reads are more than enough.
20 million. Okay, I understand.
Yeah. So you don't need to have too much, because the main problem when you're doing the like this, because when you collect in the cave, okay, so you don't have a impurity. But when we collecting our like feces, because feces almost 50% DNA comes from the human cells, and because this is like you have to remove when you analyzing your data. So if you don't remove it, then you always end up like less. Then if you don't have enough like kind of a diversity, then their data is not kind of reliable. So we prefer to have around 50 million. So then we remove almost like a 30 million or 25 million from the human samples. And then we get rest 20 million, which is like good reads. And sometimes other impurity because the, and so minimum, I think you have to have final like a 20 million reads, the pure, then it's fine. In your case, I think 20 million reads should be more than enough for the.
And regarding the number of patients, I don't know, 10, 20, 50.
Yeah, this is always a difficult question because there's no specific number because I mean, statisticians decide, okay, whether the p-value or whatever means I'm not very fan, but I try to understand the biological phenomena because how much biological phenomena you can understand sometimes has a trend, or maybe the significance is not there. So this is because the human, like the patient we have, like is always a lot variable. Each person has a different genetics, and getting a like a very concrete cohort and the same time and everything is very impractical because even the PD patient comes, somebody has a drug, somebody has a different genetic makeup, and it's always kind of difficult. So we try to do like if we taking say like Parkinson patient, we try to keep as a control from their partners, like male or female. But that's again the female has a different microbiome, so this is it's always difficulty in experimental design, which one we should consider as a proper control because nobody is properly healthy. There always somebody we say like, okay, this is like diseased and non-diseased. This is with respect to this particular disease. Then we say like control because there's no healthy such control exist in the like in anywhere.
Thank you very much.
Okay, thank you.
Thank you, Yogesh. Any questions from the audience? Actually, I have maybe, because in a certain moment, Yoga showed in the presentation that there is kind of fecal transplantation to
Yeah.
modify the microbiota. So I don't know if you got it. So we transplant fecal material. So how it works, and this is the first question. So I don't know. So how it works, and if people, patient, really accept that.
So I think there's like not many studies. So I means because of time limitation, I removed like few of the slides earlier because so what happens normally when you have a very complicated inflammatory bowel disease? So when the nothing is working, so then people kind of volunteer because they want to have some kind of help. So then they normally look for some healthy person from the same family because genetics also matters. And so then they just have like they took the fresh pieces and then they just remove all the like particulate material. And then they normally, I don't know whether you know any like means anemia, so they do to the anal transplant. So they cannot go through oral, so the anal transplant, and then they give like a certain, and the effectiveness is around 35 to 40%. And because they have say presented like 10 individuals, which is inflammatory bowel disease, only like a 3 or 4 responded, and most of them not responded. So always these the clinical study always you have to not go on the main heading. You have to go in between the lines what actually they have found, and that's what I talked earlier, like how stable the microbiome on longer term, because they give like up to like talked about 6 months studies, but not long-term studies. But animal studies, yeah, we know a lot because animal studies we can like track the animal cell up to like 6 months, 1 year. But animals are not the same like patient. But I mean, this is a kind of thing that planning, and that's why people are thinking of using the synthetic microbiome because synthetic microbiome eliminates all this like a genetic kind of makeup. Because if you know the community, what kind of community would be, the synthetic microbiome is the way forward for the future because then people will easily accept. Because if you say think, some people are thinking forward, but some people don't want to take maybe feces because they think that there could be different like context, like cultural things and so on. This is difficult aspect.
Of course, of course. So, Yogesh, yes, please.
Thanks for this nice talk. You said in your presentation that the high diversity of species in the gut microbiome.
You hear?
No, I think you have to repeat the question. That's the online.
Thanks, Yogi, for this nice talk.
Thank you.
You said in your presentation that high diversity of species in microbiome gut is an indicator of good metabolic waste and good health. You are sure? I don't know, but I think that the problem is not the problem of number, but it's species. Is the quality of species in microbiome is an indicator of health or bien of good metabolic waste.
May I agree with yeah, I agree with you because more the diverse is like alpha diversity, as Latif talked earlier. So that means this is kind of a more important aspect. But having a ratio is not the good indicator because this is like in 5 or 10 years back is considered more kind of okay. If you have very high ratio of Firmicutes and bacterial ratio, then maybe you're more prone to develop like obesity or so on. But this is not very considered now anymore gold standard. This is kind of I think all the scientific community trying to eliminate. So mostly like trying to fix the like more the diverse community of the like species you have in your colon, the more likely you will have a more kind of a good, kind of consider as an indicator of a good health. But still, I think a lot to be done because this is always like the studies has been done on a certain population, and then I think once we have a whole like kind of a different countries different, then you can think about like maybe it could be country specific or could be like particular environment specific. So these things actually has to be addressed in the future as well to really say, okay, if we have a really good microbial diversity is the good health indicator. So this is what I would kind of phrase this way.
Okay, thanks.
Thank you.
Yogesh, again, thank you very much. It was very,
Thank you, sir.
I was very happy to see you again after all this year, and wish you best of luck. And maybe I will see you tomorrow with the Madi talk.
I think, yeah, maybe I will not be attending, but she will be joining later on.
Thank you
Okay,
very
then thank you very everyone for listening, and then hope I try to convince some, give you a bit more knowledge. Okay, then bye bye.